Hello friends, today we are going to study about nucleic acid part 4 and in this video we'll study about Syn-Anti conformation, sugar-Pucker conformation and different conformation of DNA helix along with some unusual structures. Now first, Syn and anti conformation. What do we mean by Syn and anti conformation? Now this, as you know, is a structure of adenine. Here from this N, it is connected with ribose sugar. This conformation is known as anti-conformation because here this benzene ring of the adenine is on the other side of this ribose sugar. Therefore, this conformation is known as anti-conformation. Coming to syn conformation, how would it be? This is ribose sugar. This conformation is known as syn conformation because here the benzene ring is just above the ribose sugar. So when the benzene ring is just above the ribose sugar, it is known as syn conformation. When the benzene ring is not above the ribose sugar, it is on the other side, then we known as anti-conformation. Coming to cytosine, which is a pyrimidine. Now here, from this end, it is connected to ribose sugar. This conformation is the anti-conformation. The conformation where this oxy group or this oxygen group of the pyrimidine is on the other side of the ribose sugar. This conformation is the anti-conformation. How its syn conformation would be? This will be the syn conformation where the oxygen is just above the oxygen of the ribose sugar. This conformation is known as syn conformation. So in case of purine, the conformation in DNA can be anti or it can be in the syn form also. But coming to pyrimidine, the conformation should always be anti. Why? Because in case of syn, this oxygen will come close together and because of which they will repel each other, which will disturb the DNA structure. So in case of pyrimidine, it will always be anti-conformation so that the oxygen of ribose sugar and the oxygen of the cytosine should be placed apart. Same will work for the thymine also. Here in thymine also you can see here it is oxygen group. So in case of syn, this oxygen will come just above the oxygen of the ribose sugar. So in case of pyrimidine, whether it is a cytosine, whether it is a thymine, whether it is a uracil, even in uracil also in this position oxygen is present. So therefore, for purines, it can be in syn or anti form, but for pyrimidines, it is in the anti form. Same for guanine also because guanine is also purine. So it can be in syn or anti form. Next is sugar-Pucker conformation. The sugar in case of DNA or RNA, it is a ribose sugar. This how we draw the structure of sugar. This is first carbon with which base is attached. This is fifth carbon. This position is third position and this is the second position. So in case of sugar-Pucker conformation, it can be in one of the two forms, that is, if the third position or the 3' position is in the direction of 5' position. For example, this is 5' position. So this 3' position from this end, it shifted inside towards the 5' end. So this is known as 3' endo conformation. These positions, which is normally we draw the ribose sugar, these positions 3' and the 2' are in the opposite direction of 5'. So these positions are called exo. 2' exo position, this is 3' exo position. But in case of 3' endo position, the 3' end or the third carbon will be towards the 5' end or the fifth carbon. Similarly, if we need to draw the 2' endo, this is the 2' endo position. So in case of DNA is a very flexible molecule. The sugar phosphate bonds, all the bonds of the ribose sugar, the sugar and phosphate bond, these are very flexible. They are not completely static in one form. So they move. The bonds are very flexible. So they take these type of conformations. That is 3' endo or 2' endo conformation. So endo means inside. Exo means outside. So normally if when we draw the ribose sugar, so this is 2', H or OH in this side. So this is exo. Here in 3' also OH is in outside. So it is exo. Opposite direction of this fifth carbon. In case of endo, if we are saying 3' endo, that means 3' position will be towards the direction of fifth carbon. Similarly, in case of 2' endo, the 2' position will be towards the direction of the fifth carbon. So these are the sugar-pucker conformations. Now there are different conformation of DNA helix: A form, B form, and Z form. So here all the properties are given and the difference between all these forms are given. Importantly, I want you to note that in case of A form, glycosyl bond conformation is anti. For B also anti, but in case of Z, cytosine is anti, guanine is syn. Next, A form is mainly observed in whatever Watson-Crick model of DNA we studied, that is the B form of DNA. A form of DNA is mainly found in RNA. RNA, though it is a single-stranded molecule, but wherever the complementary sequences are present nearby, palindromic sequences are present nearby, they fold. So when they take a fold or when they take a secondary structure, they mainly form helix type, that is A type. And in case of DNA also, sometimes certain DNA protein complexes, for that binding, the DNA takes A form. So when there is low humidity, the DNA takes the A form, or the A form is more prominent. In case of high humidity, which is normally the case in the cell, because cellular environment is aqueous environment, so in case of high humidity, mainly the DNA takes the B form. Z form, because it is antisyn, Z form means what? Zigzag, zigzag. So in case of Z form, the bases take anti, then syn, then anti, then syn, and when it will happen? It will happen when it is purine pyrimidine alternative mainly. And when it happens, when mainly in a DNA structure, we have a long stretch of purine and pyrimidine alternatively. In that case, we'll get a ZDNA, and that too when there is high salt concentration. So these are the few things to focus on. Rest all the properties are given in the table. Now there are some unusual structures. These unusual structures are not found in the complete DNA strand. It is a sequence dependent structural variation. That means whenever that particular stretch of sequence is found in any particular segment of the DNA, that segment will take this unusual structures. In that there are two, that is bends and hairpin. Bends means whenever there is adenosine stretch. If 3 to 4 adenosine stretch is there, the DNA form bends at that site. So the 6 adenosine in a row produce a bend of about 18 degree. So DNA will bend like this wherever there will be minimum of 3 to 4 adenosine residues in a sequence. So these bends or hairpin, these are very important during gene regulation. Because of this bends, proteins can able to bind with that particular section of the DNA. What is hairpin? Hairpin means, for example, in this stretch of DNA, suppose the sequence is present. After few nucleotide gap, the sequence present is like this. So what will happen? They will form hairpin like that. So this is called hairpin. So whenever there is a inverted repeats or the palindromic sequence in a DNA, in a single strand of DNA, it forms the hairpin. Now when this happens in double strand, actually DNA is double stranded. So in this double strand, when like this hairpin forms, that means each strand, because of this inverted repeat, are paired among itself. So this structure is known as cruciform. If it is in one strand, we call it as hairpin. If it is in both the strand, we call it as a cruciform. These are the unusual structures found in the DNA, not in the complete stretch of DNA, but wherever we'll find such sequences or patterns. Now one is triplex DNA. Triplex DNA means like this. If you see, this is thymine, this is adenine. So here, this middle A is forming bond with this T. This is the normal bonding. But the another T in the close vicinity come and bind with this adenine also. So this forms the triple DNA. Similarly here, CGC. So here what happened? The G in the center, from both the side it is bonded with C. But this C in the black, this is the normal pairing which should happen. But this C in the pink color, it forms double bond with this G. So it is not the normal pairing. If you see the structural difference, what you can see? This point, NH2. is forming bond with guanine. Here hydrogen donor. Here also you can see NH2, hydrogen donor, forming the bond. Then the middle N forms the bond with guanosine. This one. Here also the middle N is forming the bond with guanosine. But can you point out the difference in both the cases? The black and the pink. Here the donor of hydrogen is guanine, NH. But here in the left side, the donor of hydrogen is cytosine. So in this case, in the cytosine, the extra H comes and bind, and that's why it can behave as a donor, which was not there in the normal DNA molecule. Understood the difference? In the normal DNA, here the guanine is the hydrogen donor and not the cytosine. But in case of this pink cytosine, the cytosine is the hydrogen donor and not the guanine. That means the nitrogen of the cytosine become protonated and hence can act as a donor. So this is not the normal pairing. This GC pair is the normal pairing, which is known as Watson-Crick pairing. But this is not the normal pairing. This is known as Hookston pairing. And this happens when the pH is low. When the pH is low means H plus is more. When the H plus is more means more chance of getting protonated. Low pH, more of H plus. When more of H plus is present in the surrounding, so more H plus will bind with the cytosine, making it the protonated molecule and forming the triplex DNA. So this unusual pairing is known as Hookston pairing. The triplexes are stable at low pH, and these triplexes are readily formed within the long sequences that contains only purines or only pyrimidines. Like triplex, this is G tetraplex. It happens whenever the guanosine residues are present in a long stretch. HDNA means it happens also in a polypyrimidine or polypurine tract. It mainly happens when there is a long stretch of GC repeats. So what happens is, if these are the two strands where like this repeats are there, these strand will fold. Maybe here also some like this the repeats are there. So what will happen? Here also they'll form the bond. With this also it forms the bond. So such structure is known as HDNA. This also happens when there is a long polypyrimidine or polypurine stretch. So these are not found in the entire DNA length. It only found wherever such sequences are dominant. Now one question from my end for you all. When sequence is given, you have to write the complementary strand sequence. And also you have to tell what special type of sequence is contained in this DNA segment. And because of that special kind of sequence, what alternative structural arrangement that DNA can take. Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.